So today we'll start the chapter Microbes in Human Welfare. In human welfare. Okay, this is a very easy chapter, but in this chapter you cannot skip a single word even. Okay, so as usual I am giving you point wise detailed notes, and no word or no line is missed by me here in this will be missed by me. And again you are requested to write the notes and go through it. Then whatever homework will be given, please solve those homeworks without taking the help of Google or ChatGPT. Okay, you know what are microbes? The microbes are the organisms which cannot be seen through naked eye. They can be seen under microscope only. Okay, so these microbes form major component of the biological systems along with the plants and animals. Plants and animals are the major components, but besides that the microbes forms the major component, one of the major components of biological system on the earth. And these microbes are extremely abundant and diverse. There are varieties of microorganisms. Okay, now let us see how the microbes are indispensable components of the ecosystem. So microbes are indispensable components of ecosystem. The ecosystem performs certain functions, and these functions cannot be performed, cannot be completed without the microbes or the microorganisms. And these microbes are found everywhere. They are found in soil, water, air, human body, animal body, plant body. So everywhere they are present. Okay, now these microbes are also seen in extreme conditions, like they can be seen in the thermal vent or in the deep freezing point also they can survive. There are some microbes which are surviving at the freezing temperature also. Okay, so microbes in extreme conditions. Okay, for example, they can be seen in deep inside gaseous, that is, you can say the thermal vent, means the outlet where very hot water is released, that is called thermal vent. Then even they can live at temperature as high as 100 degrees Celsius. Our body temperature is 37 degrees Celsius, and we can withstand maximum up to 50 to 55 degrees Celsius. But these bacteria or you can say the microorganisms can survive even at 100 degree Celsius. They can survive deep inside the soil. Okay, they can survive without oxygen. Not all microorganisms. Some of the microorganisms are anaerobic and they can also survive under thick snow layer, highly acidic environment, so like this they can be available in varieties or diverse environment, and these environments are so harsh. Okay, so microbes show extraordinarily adaptability to extreme environmental conditions. From this, what is the conclusion? The conclusion is that microbes show extraordinarily adaptability to extreme environmental conditions. Okay, now what are the microbes? In class 8th I think you have studied about the grouping of the microorganisms. The microbes are grouping of microorganisms. They are grouped as protozoa. It includes which are unicellular, eukaryotic organism. Okay, the next is bacteria. They are prokaryotic, okay, and having different shapes that you have studied in class 11th. What are those shapes? They may be rod shaped, they may be spherical, comma shaped, or they may be spiral. Okay, so they may be of a different shape. Then the third category is fungi. The fungi are eukaryotic and heterotrophic, and they include the yeast and mold. Okay, then another group of microorganisms which are not actually considered as microorganism, that is the separate group we can say, that is the viruses. Okay, so these viruses are microscopic infectious agent. They are microscopic infectious agent. Okay, so here I have not written them as organism. Okay, and there are different types of viruses, like for example, if they infect the bacteria, then they are known as bacteriophage. Okay, so they infect bacteria. Similarly, your animal virus and plant virus means they infect the animals and plants. Besides this, some other microorganisms are there, that is that comes under the virus only. They are viroids. So what are viroids? Viruses are the infectious RNA particles. Remember, RNA particles lacking protein coat. A bacteria is having a protein coat and the genetic material, that may be RNA or DNA, but the viroids are only infectious RNA particles. They don't have any protein coat. Okay, the next one is prion. You have already studied this in class 11th. So these are infectious proteins, and these are only proteins. They don't have without nucleic acid. Okay, so they don't have RNA or DNA, only the proteins are there, and these proteins are infectious. Okay, now all these microbes can be grown in culture media. Now let us see what is culture media. The microbes can be grown in culture media. So what is culture media? That is nutrient media. Culture medium means nutrient medium. Now these organisms, when they are grown in the nutrient medium, they form visible colonies. Okay, so these microbes are invisible. They can be visible under microscope only, but when they are grown in the nutrient medium or culture media, then they form colonies which are visible. Okay, now these colonies, what are these colonies? Let us come to colonies. Means the visible masses of microorganisms growing on nutrient medium. Okay, and what is the use of forming these colonies? These colonies are helpful, one, for microbiological studies. And other one is, it is very helpful in identification and research. Helpful in identification and research. That means the first one is microbiological studies. You cannot study the microbes under microscope always. Suppose that you want to study the behavior of the microorganism, its structure, then you have to grow the culture. grow them in the culture, and after getting the colonies only you can observe their behavior. So that is the microbiological studies. And besides that, we can study their genetic material, their structures, their function, everything in the colony only. Okay. And these also formation of colony is also useful in research work. Okay. Now these microorganisms are not always harmful as we think. Okay, it is a myth. The microorganisms are both useful and harmful. So today we'll discuss about the usefulness of the microbes, because the chapter is microbes in human welfare. In what way the microbes are useful for us, that we will discuss. Okay, so we have heard about the diseases that is caused by the microbes, but all microbes are not harmful, and there are many microbes which are very useful for the humans. Okay, so that means microbes have both beneficial and harmful role in nature. Okay, so today we'll discuss only the usefulness of the these microorganisms. So the first topic is microbes in household product. Okay, so let us see how they are useful in household product. The first one is, we'll discuss one by one. The first one is curd formation. Okay, the curd is formed with the help of the microorganism, that is lactobacillus and or you can say the lactic acid bacteria. So the microorganisms involved are lactobacillus, or other lactic acid bacteria, that is lab, LAB. Okay, now how the milk is converted into curd. So I am giving you the schematic representation of this one. So please, this question comes in exam, then please write in this format only, and nobody can cut your mark. Okay, so now process of conversion of milk into curd. Okay, so let us see. Lab grow in milk. Lab means what? Lactic acid bacteria or the lactobacillus. So this can be lactobacillus, or the lab is grown in the milk. Then this lab produces acids. Okay, and this acid coagulates the milk protein. Okay, and now the milk is converted into curd. Okay. Now what is coagulation of milk? So let me write down here only. Coagulation of milk means conversion of liquid milk proteins into semi-solid form. Okay, so this is called coagulation. Now, here what happens, the protein, milk protein is not completely digested. The milk protein is partially digested. Okay, and how it is helpful? How the curd is helpful? So, helpfulness, it's lab digests the milk protein partially, and this increases digestibility. Okay, if you take the milk directly, then it is very difficult. It will take longer to digest the milk protein. But if you take the curd, then the milk protein is already partially digested by the bacteria. So very easily it can be digested in the in our digestive system. Okay, so now this curd can be used as a starter or inoculum. It is used as a starter or inoculum. So this is helpfulness of lab. Some other nutritional importance is also there. So let us see. The curd is used as starter or inoculum. Very important. Okay, that means small amount of curd is added to the milk to form the curd. So little amount of, you might have observed at your home, your mother adds a little amount of curd to the milk so that the next day the curd is formed. Okay, now what is the importance of this starter or the inoculum? Okay. Importance of starter. What is the importance? It contains millions of lab, and this lab multiply rapidly at suitable temperature. So suitable temperature means bit warm temperature. Okay, and milk converts into into curd. Now, what is the nutritional importance of curd? Okay, so the lab in the curd increases nutritional quality by increasing vitamin B12 content. Okay, and this also, it also is the role of a lab that is done in the stomach. Lab also suppresses disease causing microbes in our stomach. Okay, and it also maintains healthy microbial balance. In the stomach. Okay, so the lactic acid bacteria are beneficial microbes present in both curd and the human intestine. Now the next use of microbes is fermentation of dough. Dough, you know, the batter. Okay, and this dough is used for preparation of dosa and idli. Okay, so this dough is fermented by bacteria and this, because of this fermentation, carbon dioxide is released. Carbon dioxide is produced, and because of this production of carbon dioxide, two things happens. This is carbon dioxide. Okay, because of this production of carbon dioxide, two functions are accomplished, that is, dough become puffed up. Okay, and the texture becomes soft and spongy. Texture become soft and spongy. Okay, so this puffed up appearance of dough is due to release of carbon dioxide during fermentation. This is very, very important. Now, what is the metabolic pathway involved in this process? The metabolic pathway is fermentation. Remember, fermentation is the metabolic pathway. Okay, and these fermenting bacteria are naturally present in the environment and the ingredients. Then the next use is bread making. So the organisms used are Baker's yeast, and that is also known as Saccharomyces, cerevisiae. Okay. Now, what is the process involved in it? Process. Yeast ferments the dough. Then carbon dioxide is produced and the dough rises. Okay. As a result, bread becomes soft and porous. Okay. Now there are some traditional fermented products obtained due to these microbial activities. Let us see what are they. Traditional fermented products. So they are used in the preparation of traditional foods and the drinks. For example, toddy is a traditional drink of southern India. Okay, and it is obtained by fermented sap from the if the palm tree is cut in a particular region, some fluids are released, and these fluids are the sap, and when these saps are fermented, it forms the toddy. Okay, and the other fermented foods are fish, soybean, and bamboo shoots. Okay, then the next one is cheese production. So this is one of the oldest microbial food product, and there are the cheese is famous because of its texture. There are different types of cheese depending on its texture, flavor, and taste. So varieties of cheese are due to different texture, flavor, and taste. Okay, so these characteristics depends on microbes used. Okay, so for example, your Swiss cheese. Swiss cheese, the large hole, so here the texture is different. The large holes are due to a large amount of carbon dioxide produced by a bacterium that is called Propionibacterium. Propionibacterium. You have to underline it because it is the scientific name, Propionibacterium cermani. Okay, so you have to follow the rule of nomenclature. So the large holes in Swiss cheese are due to the carbon dioxide produced by the Propionibacterium cermani. The next is Roquefort cheese. So this Roquefort cheese is prepared by, it is ripened by using a specific fungi. Okay, and which provides characteristic flavor. Okay, so these are important for the flavor of the Roquefort cheese. So this is about details about the role of microbes in household product, and in our next class we'll discuss the use of microbes in other fields. For today, this much only. Thank you.